Good morning, my dear students. Today we are discussing about serological and immunological tests. So, serological and immunological test it is a study of the blood serum. So, serological test is a study about the scientific study of the blood serum. Study of blood serum. Scientific study of the blood serum. So in practice, the term usually refers to the diagnosis and diagnostic identification of the antibodies in the serum. So indirectly, it is the diagnostic identification of antibodies in the serum. So the serological and immunological tests are involved in the diagnosis and identification of the antibodies in the serum. And the next one, such antibodies are typically formed in response in case of the antibodies are presented in the serum, no problem at all. In case of typically formation, typically formed in response to infection, response to an infection. So in infection, so in case of easily identified in the serum, that person is going to healthy. In case of typically formed the response of the antibodies in the serum, that is called as the infections may occur. The infections is with the microorganisms in other foreign proteins and any other substances are entering into in our body. There is no chance for the identification of the antibodies in our serum. So that indicates the infection. This serological test may be performed the which purpose we are using. So the serological test may be performed for diagnostic purpose. For the which purpose? Number one, infection suspected purpose. Infection is suspected purpose, and rheumatoid illness and diagnosis of the rheumatoid illness. Rheumatoid illness means arthritis related diseases. And number three, it is a checking of the blood type. Individual blood type. And the next one, serological test help to the next one, the serological helps to diagnosis of patients with certain type of immune deficiencies. So it is the helps to the diagnosis of patient with certain immune deficiencies. Certain immune deficiencies associated with the lack of antibodies. So in case of any antibodies are decreased in our body, that immune system is determined by the serological test. And the next one, several serological techniques that can be used depend upon the antibodies being studied. So serological tests also depends upon the antibodies. So that are the including number one, ELISA, enzyme linked immunosorbent assay, ELISA, agglutination, precipitation, the next one, complement fixation, complement fixation, and other fluorescent antibodies. Fluorescent antibodies. These type of reactions are depends upon the antibodies studies. Depends upon the antibody studies. These are all are the detected by the serological tests only. And the next one, some serological tests is not limited to blood serum, but can also performed in other body fluids such as saliva, and the next one, similar properties to serum. Saliva and similar properties to serum, saliva, semen, and etc. etc. So serological test may be also useful for the preparation of the pieces of the evidence purpose also. It is a study of the study purpose. Which study purpose? So evidence purpose. It is the study the evidence purpose. Evidence. So it is used in the study for the evidence purpose in the at the moment of rape conditions. So at that moment also we are determining the identify the particular semen and identify the candidates with the using of the serological test. The serological test is not only determination of the particular types of antibodies, it is a determination of the different types of tests also determined with the using of the serological test. So the serological test is it is the development in the rapid techniques and diagnosis of disease, microbes, and even illegal drugs also is determined by with the using of the serological and immunological techniques. So in the serological and immunological techniques, so first one is ELISA test, and the next one VDRL test, and viral test. Viral test, VDRL test, viral test. So in ELISA, the video is forward into our group and VDRL test. Now we discuss about the VDRL. So VDRL means here venereal disease research laboratory test. Venereal disease research laboratory test. So it is it is mainly used in the determination of the treponema pathogens. Treponema pathogens are the determined by the VDRL test. So the VDRL test is it is a serological test. Put subsiding VDRL and write down the abbreviation, and the next one main use. So main use the determination of the treponema diseases, and here the VDRL test, it is serological screening test, and it is determination of the treponema syphilis also. And used to assess the response therapy. And the next one, it is a detect the CNS involvement and diagnosis of the congenital syphilis. And the next one, it is a determination of the not only syphilis, it is a determination of the involved in the it is a determination of the different types of viruses. What are they? It is different types of viruses. And the next one, and determination of the viruses and drugs and pregnancy, rheumatoid fever, rheumatoid arthritis, and leprosy. These are all also determined with the using of the VDRL test. And the next one, viral test. What about the viral test? Viral test. So the viral test, it is the most utilized diagnostic test for the determination of the typhoid fever. This test is demonstrate the presence of the osmotic and flagellar agglutinin and salmonella type patient serum is suspected the agglutinins also. So here these are the salmonella type. It is the determination of the different types of typhoid fever. Different types of typhoid fevers also determined by the viral test. So the viral test, it is agglutination technique. It is determined. This tube is the agglutination technique. With the using of the agglutination testing, we are determining the different types of fevers like typhoid fever, different types of fevers also determined with the using of this particular technique. So this is about the viral fever, and here write down about the pregnancy test. Write down about the pregnancy test and sick test, and dick test, and the next one tuberculin skin test, and the next one lepromin test, and the next these type of test write down. This is your homework. So this is the serological and immunological techniques. So sick test, dick test, and tuberculin test. The next one tuberculin skin test, and the next one is the lepromin skin test, and these type of tests are your homework. Write down what is the, just write down what is this test name and what is the main use, and here also write down. This is your homework. If you have any doubts from this topic, tomorrow we will discuss. Thank you.